Okay, let's hope I'm going live. Can you see me? Excellent. Can you see me, everybody? And hear me? Could you just give me a thumbs up if you can hear me and see me? That would be lovely. Okay. Hello, everybody. Are you there? Can you hear me? Yeah, brilliant. Thank you. Fantastic. It's nice to have somebody communicate with me. Well, welcome to my first GCSE biology drop-in session here live. And please let me know in the chat how things are going. Yeah, brilliant. Thank you. Welcome to you all. I hope you're all keeping well. We're just waiting for the last few people, although I should think most people have now joined the drop-in session. So I hope you've all had a good day today. I'm looking a little bit windburnt. I've been outside quite a long time today doing some work on registering with my pupils for the lateral flow this week. So they all had to come into school today to get their lateral flow. So we will get started in a minute. All right. During the drop-in session, please do make comments and ask questions. You too as well, Amy, huh? Yeah. So you were getting yours done today, were you? I hope it went well. And wasn't too stressful. I know a lot of my students were a little bit worried about it, but when I'd explained them through and we'd gone through the step by step, they felt a lot better about it. And next week, they're all going to be doing it at home. So that'll be all interesting to see. Yeah, a bit scary. I know, I feel that, you know, everybody's being a little bit like that at the moment. I don't like the test because I don't think anybody loves shoving an extra long cotton bud down their throat and up their nose. I mean, I don't think anybody's going to enjoy that, are they? Yeah. So we're only a small school, so we managed to get the whole of the school through today because we're only just over 300 students. So that was all right. So I should think it's a lot harder for the big schools. Yeah. Right, so I think what we're going to be doing is I'm going to start sharing my screen with you in a minute. So I'm just getting my pad together so that I can write. So I'm going to go to share screen and see, hopefully it will all run smoothly tonight. So here we go. Just about to share my screen with you all. Right, I should have been sharing my screen. Can everybody see it? Right, yes, well, GCSE, IGCSE, it doesn't matter. My students actually do IGCSE and it works for them as well. Right, yeah, okay. Good, I'm glad you can all see the screen. So this is a drop-in session today. And I'm just going to start the PowerPoint. I hope. There we go. Hopefully you can see the welcome screen. So we're going to be looking at magnification and size today. We're going to work through some exam questions and we'll also hopefully also get on to leaf structure and function if you would like to. So do ask questions as we go along. We're doing these topics because these are topics that people have asked for during the week. So we've got magnification and size and leaf structure and function. First of all, in case you haven't actually been to any of my sessions yet, I'll introduce you to myself. So I am Philippa. I'm the lead GCSE biology teacher here at Snap Revise and head of biology at my school. I have a bachelor's degree in animal science, which is a sort of agricultural animal degree. And then I went on to PGCE in biology and games. And both of these I read at University of Leeds because I enjoyed it so much. I've been teaching and tutoring for over 25 years. I've also been an examiner for several different boards at all levels from Key Stage 3 upwards. I've written for various academic publishers. I've got used to writing sort of past exam questions and mark schemes for them in particular. So I do hope my experience is going to be useful to you. So, okay. I love my job, and I really hope that my enthusiasm for the subject at least partly rubs off on you. And I'm starting to get used to doing these live YouTube sessions as well. Okay. So to start off with, I know some of you already know about this because you've been to some of the other sessions. But for those that are new to all of this, I just wanted to explain about the GCSE tutoring, which is a new platform which Snap Revise has just launched. So Snap Revise provides these sessions. We are running on YouTube, but there's an extensive platform full of resources for you tailored to both GCSEs and A-levels. And we've got lots of new additions, including this GCSE tutoring platform covering the full curriculum. So classes each week work logically through the curriculum, fine-tuning all the skills needed to achieve your potential in your exams and your assessments. Okay. There are high-quality handouts of the PowerPoint slides used in the classes, written to help you with your notes and learning. And there's recordings of previous lessons, which are available to you in case you want to rewatch them, because we all have topics that we find particularly difficult, we might want to recap over, and it's quite useful to play them, stop them, go back over areas that you're still struggling on. It's certainly a way that I would be revising. And there's also one-to-one support for you where you can ask questions about specific areas and you're not sure about. So, and just so you know, there is this 2-week free trial. If you're interested in trying out this tutoring platform, there's a 2-week free trial. Just sign up to it on the website. You enter this code at the checkout and you can claim the trial. So this Tutor 14, if you can see the code here, the Tutor 14, I'm just going to get my pen and it's going to, there we go, I'll go back. It seems to always do that when I go for the pen for the first time. So Tutor 14 here is the code that you need to put in, and you just go to our website, snaprevise.co.uk, click on the tutoring on the navigation menu, create an account, add the subjects you want to trial, and use the code at the checkout for your free access. Okay, yeah, we're going to start now. It's just so that people know. So to start off with, I was going to talk to you about magnification. So that's the start of the session, is what we're going to look at is magnification, because you all need to know how to use microscopes, don't you? And we can use microscopes to examine the details of cells and their structures much more closely. So at school, you would use a light microscope. And on the light microscope, you have an eyepiece and an objective lens. So an eye lens and an objective lens, don't you? And those multiplied together give you your magnification. All right. So on your school ones, if we want, we're going to be going straight on to questions after this. I just want to go very, very quickly through the start of it. So the eyepiece is normally times 10 at school, and your largest objective lens is normally about times 40. So that will give you an overall magnification of type 400. And the actual size of the cells and structures can be calculated if we know the image size and the magnification. So we can actually work out how big a thing is in real life by measuring the size of the image that we can see, and we know the magnification that we've used. And so we need an equation for this. So do you know what the equation for this is? It's often seen in a triangle like this. Yep, okay, yeah. So we can do, I always remember it as I am, because I am brilliant at biology, or I am able to do this magnification. That's how my students remember it. So it's the image size. So that's what you're actually seeing. This is the actual size of the real structure that you're looking at. And this is the magnification. So I always remember that as I am. All right, I am. And then hopefully that will help you. And so obviously to find the actual length, so if you want to find the actual length, you divide the image by the magnification. If you want to find the magnification, you divide the image by the actual. If you want to find the image size, you multiply the actual by the magnification. And so there we go. Now, we will be looking at this in a moment, but to actually, in an exam question, you might either be given the actual size and you've got to work out the image size, or the other way around, or you may be given a scale bar where you've actually got, say, a cell like this, and then it's got lines down here, and this is a scale bar, and this is drawn onto the diagram to show where it has been measured from and to, and or it might just show, it might give you an actual size that the person has measured, say on their diagram it's 4 centimeters or something like that, or it might actually have a ruler next to it with the measurement on there, and you have to work out the size of it from that picture. So that's using a scale bar. All right. So moving on then. So that's just a rough overlook of the basic stuff that you need to know to complete a lot of the exam questions on magnification. So an awful lot of it is about just doing the maths when it comes to the magnification. So let's have a look at our first exam question then. So here we've got, they've actually taken a picture of Vibrio cholerae, which are bacteria that cause cholera. And this one has a magnification of 8 times 800. And it's then telling us that the length of one flagellum is 68 millimeters. So it says, calculate the length of the flagellum in micrometers. Now, this is the area where people sometimes get stuck, is making sure you use the same units of measurement for the actual and the image that you see. All right, so let's see whether any of you can work this out. So we've got the length of the flagellum is 68 millimeters. So the length of one flagellum is 68 millimeters. And the magnification is 8,000. Brilliant, well done. So we've got the remembering I am, we have got the image size equals 68 millimeters. We've got the magnification equals 8,000. And so the actual equals 68 over 8,000, which equals, yeah, brilliant. So it starts out at 0.0085 millimeters. How many micrometers are there in a millimeter? Can you tell me? Yes, Evan, you've done it the other way around, which is absolutely fine. Some people prefer to multiply it up at the beginning. Some people like to work it out at the end. All right, brilliant. Good. 1,000. So there's 1,000 micrometers equals 1 millimeter. And so we multiply this 0.0085 by 1,000. 1, 2, 3. And so we end up with 8.5 micrometers. Good, well done. So you'll get full marks for the correct answer without even showing you're working on this one, because it didn't ask for working. So if it asks for you to show your working, you need to, but this one didn't ask for that. All right, well done. So let's move on to the next one. Okay, so this one is looking at the cells in figure 3 are drawn to a length of 100 millimeters in the student's textbook. All right. The actual length of the animal cell is 60 micrometers. Calculate the magnification this time to 2 significant figures. So again, we've got our I, A, M, I am, and that's where you've got to keep telling yourself, I am brilliant at biology. All right, so the image we've said is 1,000 millimeters, and the actual, so who is going to come up with the answer for that? Okay, brilliant. So we've got, so again, you can either work it out in millimeters to start off with, or you can work it out in the micrometers. It's up to you, whichever way you prefer to do it. So a lot of you are liking to multiply it up, so we'll do it that way this time. So 100 millimeters is 100,000, isn't it? And it's over the actual, which is 60. And so that gives us, yeah, you've come up with 1,700. So it comes up with 1666.67, 2 significant figures, but to the nearest whole, it is 1,700. Okay, well done. So show clearly your marking, here it says show clearly how you work out your answer. So here you would need to give your answer. Yeah, this one is to 2 significant figures, to 2 decimal places, sorry, 2 decimal places. This one is to 2 significant figures. All right, well done. Excellent. So moving on then, let's have a look. I think the next one we have got, yeah, so we're trying to give you examples of each of them. So this time we're looking at the scale bar. So this shows a nice diagram of a scale bar. Now, the problem with this one is they have given you the start line here, but they haven't given you the end line. So they're expecting you to use your judgment to work out where the end is. All right. So it says, calculate the actual length of structure X in figure 4. Show your working. So again, it is IAM. All right. So brilliant. So we want to calculate the actual length. So we want the actual length this time. Yeah, it's image over, yeah, so you've got your image size. So you're trying to find the actual length. So it'd be image over magnification. And we've got our magnification there. Brilliant. So we've got 100 over 500. Well done. 100 divided by 500. Okay. So, which is 0.2 millimeters. All right. Yeah, 0.19 is fine. You could have got anything between 98 and 100 here, depending on whether you went from there or whether you went from there. So they allow, if they haven't given you the actual line there, they will allow some adaptation of what you have, where you've chosen to go for the end, as long as it's as sensible. Can you do an estimate as well, or does it have to be 100? Well, like I said, it depends. You could go from anywhere from drawing a line, and I would use a ruler and a pencil in my exam to make sure that I'm not doing, if you do it freehand, you could be out by a millimeter. So do use a ruler and a pencil by the end. So it could be anything from there to the end. All right. So it could be anything sort of around 0.19 to 0.2. Okay. Is that all right? So that gives us 0.2 millimeters. All right. And if we then change, because we've started, so we've got the magnification, we've got the image. So if you wanted to put that into, that is 200 micrometers, of course. All right, because you multiply it up by 1,000. Are we okay with that? Don't worry, Shu, you haven't missed too much. We've been talking about magnification, so we've just run through a few questions on that. All right. Are there any questions about the magnification questions we've done so far? Are you finding them quite okay, are you? Thumbs up if you think you're doing all right and you're quite happy with these. All right, so yeah, some of you, brilliant. All right, so let's move on to the next exam question then. So we're looking at a cheek cell. Hopefully some of you have had a chance to look at your own cheek cells at some stage. This one has been magnified by 250 times. Brilliant, thumbs up, good, you're good, excellent. So we're looking at the cheek cell. It's magnified 250 times. The width of the cell is shown by the line D to E. So here we go, D to E. Calculate the width of the cheek cell in micrometers. Complete the following steps. So they've actually given you the steps here. Measure the width of the cell using a ruler. So obviously you can't measure this on the screen, so we're giving you it's 45 millimeters. Good. So use the equation to work out the real width of the cell in millimeters. So they've given you the real size equals the image, which is 45. And yes, you're absolutely correct. 45 over 250. So 45 divided by 250, that gives us 0.18 millimeters. And then we need to convert that to micrometers. Well done, Varya. Yeah, that's brilliant. 180, good. Yeah, there's a lot of you getting this right. Well done. Excellent. 180 micrometers. Okay. Right. So that's a nice, easy 3 marks probably for most of you. Although there are some people who are loving biology but do not find the maths side of it particularly easy. And I still have some A-level students who will forget the magnification because they haven't practiced it for a while. So it's all about just making sure you keep on top of your vision. Just make sure you keep recapping over all of your topics just to make sure you're on top of them. Right. So we're going to be looking now that you all seem to be quite happy with that magnification work. Any questions on that magnification work? All happy with that? Brilliant. Okay. So the questions that are going to be following this are going to be about the leaf. So the leaf is an organ of the plant. What is the purpose of the leaf? What happens in the leaf, actually? Let's say that. What happens at the leaf? Anybody tell me? I mean, it's designed for photosynthesis. Brilliant. So the leaf is designed for photosynthesis. That's the abbreviation for photosynthesis, PS, photosynthesis. Brilliant. Something else happens at the leaf, which helps to draw water up from the roots. Yes, brilliant, Evan. Palisade cells. So photosynthesis, we've got the palisade cells, which are packed with chloroplasts at the top of the leaf. Osmosis, yes, we've got osmosis drawing water in through the root, producing the food for the plant. That is the photosynthesis, because they're autotrophs. Okay, brilliant. Green pigment, good, green pigment in the, which is the chlorophyll. And that's in your chloroplast to trap sunlight energy. Yeah, trap sunlight energy or trap light energy. Trap light energy. Yes, there's translocation through the phloem. So we need translocation because we need to get that food away from the leaf, don't we? And that goes through the phloem. And transpiration, excellent. Transpiration, and that involves the xylem, and it involves something happening from the leaf. Yeah, transpiration due to the opening of the stomata. So we have transpiration due to evaporation of water from the leaf. All right. And that evaporation of water from the leaf helps draw water up from the roots and also helps cool the plant, especially when it's a hot day. Diffusion of carbon dioxide, excellent, yes. Carbon dioxide needs to get into the leaf. So we have gaseous exchange as well, don't we, happening in the leaf. So all these things, what affects transpiration? So transpiration is when the transpiration stream is the drawing up of the water from the roots, from the soil, through the roots, up through the stem, through the xylem, and it evaporates out through the stomata. That helps cool the leaves, and it also helps draw the water, which brings mineral ions from the soil as well. So that's really important, okay? Yes, we have active transport in the roots, active transport of mineral ions into the roots, osmosis of the water into the roots. Well, hopefully you haven't, that the osmosis and the active transport, you may have come through these, you might have heard a little bit about this when you talked about osmosis and active transport and diffusion when you started that right at the beginning of your course. Helps prevent the plant from wilting. Yes, so what happens when the plant loses too much water, it's going to wilt. And that's not good for the plant because it's losing too much water from its cells, so chemical reactions can't occur. So if they shut the stomata, which are the holes mainly on the bottom of the leaf, it will help prevent the wilting of the plant. And the things that affect the rate of transpiration, yes, thank you, are light intensity, temperature, humidity of the air surrounding the leaf, all right, and wind as well, because wind can, think a bit about what it's like when you're drying your clothes on the washing line. So transpiration is affected by all of that. Turgid, good, that's another good word, turgid, and we're going to be coming across that. So turgid means when the cell is full of water and the inside of a plant cell, and it's pushing out against the cell wall, and the cell wall is pushing back. The cell will burst if there is too much water, only with the animal cells. So with animal cells, they've only got a cell membrane around the outside of them. So what happens is if too much water gets into the cell, they swell with water and the cell membrane can't hold that cell together anymore and they burst, and that's known as lysis. But with plant cells, because plant cells have this strong cell wall around the outside, they have it to help with their structure. So they have a vacuole and the water moves into the cytoplasm and into the vacuole, and these push out, this puts force outwards onto the cell wall, and the cell wall pushes back. It's a bit like blowing up a balloon in a wooden box. All right, so the balloon fills the box, but it can't burst because the wooden box is stopping it from expanding anymore. All right, so the cell wall stops the cell from bursting. And that's really important because a lot of plants don't have any other way of holding themselves upright. They don't all have woody stems, for example, yet we have skeletons, etc., to help hold us upright. Does that make sense? Yes, the cell wall is made out of cellulose. Good. Cellulose, cell wall. And that gives it strength, so it helps hold the inners of the cell together and gives support. Good. Are we happy with that sort of overall? We've gone a little bit away from the leaf, partly away from the leaf, but not from the leaf as well. So yeah, so many things to talk about. That's the problem. So many things to talk about. So like I said, we've got the stem, the root and leaves form organ systems in the plant, and then the leaf is the main place for photosynthesis. So are there any more questions, or are you happy for me to go on to an exam question? How are we feeling about it all? So any other questions before I move on to an exam question? And you can throw the questions at me as we move through and we look at exam questions. They might make you ask more questions. So please do, sorry, you must excuse my throat. I think it's from calling out to people all day today. Excellent, move on, we will. So here, hopefully you recognize this as the structure of a leaf. So this is a cross section of a leaf. It hasn't got it has just got the vascular bundle over here. But we've got the top of the leaf here. So this is where the sun is coming down, and this is the bottom of the leaf here. So it says complete the sentence by putting a cross in the box next to your answer. So the cell that will make the most glucose is. So who can give me the letter for the cell? And do be careful, look at this very carefully. I know that sometimes people fall into a trap with this one. So look at it very carefully before you put your answer. Yes, well done. Good, Donna. And Fargia, I don't know whether that's the way to say your name. Evan, do apologize if it isn't. Amy, yeah, brilliant. So B, when it's not a chloroplast, because we're looking at the cell. Yes, it's packed with chloroplasts, but if you notice, it does say cell. So it is B. And what is the name of this? Yeah, brilliant, Evan, the palisade cell. So this is a palisade or a palisade mesophyll cell. It depends which way you've learned it. Palisade, it's all right to call it a palisade cell. So a palisade cell is packed with chloroplasts. Yeah, all of these little dots up here, those are all chloroplasts. Excellent. Because sometimes people would say cell A, but what is cell A? What have we got up here at the very top? Does anybody know what the top bit is? We've got above it is the waxy cuticle. No, that's all right. Don't worry, you weren't being rude. The waxy cuticle, so we've got the waxy cuticle, and then we've got cells underneath the, brilliant, underneath the waxy cuticle. Good, Dana. So that is the upper epidermis. So the upper epidermis, and upper epidermis is actually transparent. And the reason it's transparent is so that it doesn't stop the light getting through to those really important cells at the top, the palisade mesophyll cells. All right. The waxy cuticle. What does the waxy cuticle do? Does anybody remember what the waxy cuticle does? Be careful with your wording. To allow light to pass through. Excellent. Yeah, it's transparent to allow the light through. So transparent as well. And it allows light to, and if it's waxy, what does that do? Yeah, so waterproofing, it's not quite waterproof, but it certainly reduces water loss. There are still stomata in the upper epidermis, but it does reduce water loss. So some examples are very specific about this, that they want the fact that it reduces water loss. It also gives some protection. And the leaves in like tropical rainforests, etc., have quite thick waxy cuticles. So it helps the water run off from the leaves so they don't get weighed down by the water. Okay. Yeah, it can help protect against pest attack as well. Really good. Yeah, acts as a physical barrier, helps prevent bacterial infection and fungal infections. Brilliant. Good. Yes, that's true. Very good. So it protects against pathogens. It helps water run off the leaves so that they don't get waterlogged, they don't get weighed down by water, and they're transparent so that they allow light through to those really important palisade mesophyll cells, and it reduces water loss as well, because that is the surface, it's the top surface which is exposed most to the sunlight, and so that is going to get hotter. And so therefore, there's going to be more evaporation of water. So we want to try to reduce the water loss that is up there. All right, so we've already done cell B because that's the right one. Cell C, what is cell C? Does anybody know what cell C is? It's a something mesophyll again. And the clue is it always looks like one of these. Yeah, spongy. So, oh no. Right, I don't know what's just happened there, but I'm hoping that that is still working for you. All right, so can you still see my my PowerPoint? Because I just had a little flicker my end and my screen just went black. You can't see it? Help? Nope. Right, okay. You can't see, no. Okay, quickly, I will try and get this sorted. Right, what on earth has happened? No. Right, that's still going. That's still going. Can't, no, it's all black, technical difficulties. Right. Right, what has happened? Go live. It says that I'm streaming. Can you see me yet? Can you actually see me as a person? Hopefully. We can see you. Brilliant. Okay. So it just went black my end. That's fine. So I will be up and running again very, very soon. Play from current slide. Hopefully you'll see it in a minute. I'm sharing my screen again with you now, and you should, yes, good. I have no idea what suddenly happened there. The screen just went black. I do apologize. So hopefully now we are back to my PowerPoint. Okay, so the spongy mesophil, I've lost my annotations, but we know it's B. We know it's B, and I've got to get back to my pen. We know it's B, and with cell C, it's the spongy mesophil cell, spongy mesophil, and it looks like a sponge because it's got air spaces in it. So these are the air spaces or air pockets, and that allows for gaseous exchange. Oh, thank you. I'm really sorry about that. I really hope that there will be no more glitches. All right, so this allows for gaseous exchange. So next, I mentioned that we'd seen this over here. These are the vascular bundles. And the vascular bundles we talked about earlier were contain two tubes that are the transport system through the plant. And so brilliant, they're made up of the xylem and phloem, and the xylem carries what around the plant? So we know, so yeah, the xylem carries water around the plant, water and minerals. Yeah, brilliant. And phloem carries the food. Phloem for food. So remember the phloem and the food. Yeah, it carries some as glucose, but mainly as sucrose around the plant because that carries more. So it takes sugars. It takes other assimilates with it as well. Assimilates meaning things that can be built up into other things, assimilate them. So food and glucose. So yeah, it's the carbohydrate is normally carried as sucrose because it's unreactive and so it's easy to carry around. But it also carries things like amino acids, fatty acids, etc. around too. So it is important that we talk about the nutrients. So the assimilates, if you want to be very posh, the assimilates, which are the things that can be built up into new things. You might have come across assimilation in digestion. Yeah, it's okay to say food. It's just some people say nutrients, some say food. So phloem carries food, all right, mainly as sucrose, all right. And then right at the very bottom are these tiny pores, which we've mentioned already. What are those called? So yeah, it's phloem food. Some exam boards allow you to say food. Some prefer you to say assimilate, some prefer to say sucrose. It depends. And I also noticed that on the mark schemes, it tends to vary what they're looking for. Good, stoma and stomata. So one of them is plural and one of them is singular. So it's one stoma and many stomata. So one stoma, many stomata, and they allow gaseous exchange in and out of the leaf. All right. So they're allowing carbon dioxide in during the day when there's a lot of photosynthesis going to provide that carbon to make the glucose. Yes, and around the outside are our guard cells. I'd almost forgotten to mention our guard cells. So this is a guard cell, and guard cells are very special. We'll be talking more about those in a little while. Guard cells open and close the the stomata. So normally they open them in the morning and close them at night, but they will close them if the plant gets under water stress. In other words, it starts to wilt, as somebody mentioned earlier. The guard cells will actually shut the stomata down so that no more water is lost, because it's going to get too dangerous for the plant to lose any more water. All right. And of course, you've got the water moves out through the stomata by through evaporation, so by diffusion, because what happens is the water evaporates into this air, so it becomes gaseous water, okay, and then that will diffuse out through the stomata. So water will move out by diffusion. It's not by osmosis, because although it's movement of water, it's going through a hole. It's not going through a selectively permeable membrane. And some of my students sometimes forget that. So water diffuses out as a gas, so in its gaseous state, or as a gas. All right, diffuses out as a gas because it's moving from where it's in a higher concentration inside those air spaces in the spongy mesophyll layer to where there's less water on the outside. And that's why the more humid the air is around the outside of the leaf, less water is lost by transpiration, because there's less of a concentration difference. So all those things you learned at the beginning of biology, of osmosis and diffusion and enzymes and all of that sort of thing, you can tie them back into this topic. So you've got to remember these fundamentals when you're talking about it. Right, yes, we're going to be talking about that in just a little while, about the opening and closing of the stomata. I've run out of room on this slide now, so what we're going to do is we're going to move on to the next question, if everybody's happy with this. Yeah, okay. So moving on, guess what we're looking at now? We are looking at the stomata, okay. So looking at the stomata then, the diagram shows the underside of a leaf of a cowslip plant on a cool day and on a warm day. Complete the sentence by putting a cross in the box next to your answer. The parts of the leaf labelled X are, now look very carefully at where they are pointing to, please. So, yeah, good. Yes, the stomata. So it's pointing to the hole in the middle rather than the cells around the outside, isn't it? Good. So explain how changes to the guard cells on the warm day help the plant to survive. So what have we got? Good, well done. There's lots of you with A's there, which are the correct answers. That's really good. So explain how changes to the guard cells on the warm day help the plant to survive. So on the warm day, what's happened to those stomata? Are they open or are they closed? So just put O for open and C for closed. Open or closed, do you think, on this warm day? Okay, so yeah, they're going to be closed. Good. So if they're going to close, they close the stomata because what are they trying to do on that warm day? To prevent water loss. Excellent, well done, Janatul. So that is to reduce water loss. Good, because it gets dangerous if they lose too much water. So water is not exchanged, kind of, so water is not lost out, because we don't get exchange of water at the stomata. We get water coming up from the roots, Dana, so the roots absorb the water and bring the water up. Water is lost through the stomata, all right? So they close the stomata, that's 1 mark there, to reduce water loss, 1 mark. Or you could say to reduce transpiration rate is also fine. Reduce transpiration rate. Because what you're trying to do is reduce the number of places that the water can leave through. All right, good. So that was a relatively straightforward question. So I think we should be all right to go on to the next one, I think. Yeah. So moving on to the next one, because we've got 10 minutes left. So here we have green plants contain chlorophyll. Good, because chlorophyll absorbs the sunlight energy. The molecular formula for chlorophyll is this. You don't need to know that molecular formula. You'll be glad to hear. So plants get the magnesium that they need for the chlorophyll in the form of compounds such as magnesium sulfate through their roots. How do plants get, okay, so what, good. So how do plants get the carbon they need to make chlorophyll? So we're looking for where they get their carbon from. Okay, good. So somebody has put glucose. So they could be breaking down the glucose and using the carbon from that, or they could be putting the glucose in. Good. All right. Where else could they be getting the carbon from? Good. The surrounding air. So they could be getting it from carbon dioxide. So we've got to be specific about what it is in the air. All right. So via protein synthesis. Photosynthesis, yeah, from photosynthesis, well done. Yep, definitely from photosynthesis. So they could be taking it in from the air, from the carbon dioxide. That carbon dioxide can be used to make glucose. That glucose can then be used to build up, assimilate, build up other molecules such as this chlorophyll. So carbon dioxide gets fixed in the state of in photosynthesis. It gets fixed or formed into glucose. That glucose can then be used to build up into chlorophyll. All right, that's good. So from carbon dioxide in the air, so it would obviously diffuse in through the stomata, wouldn't it? All right. Okay, good. Yeah, people could be respiring around them, so producing even more carbon dioxide in the air around them, couldn't they? I think that's where the thought of is if you go and talk to your plants every day, then you're breathing on them and giving them more carbon dioxide. Yeah, and a product, yes, carbon dioxide is a waste product of respiration. Excellent. So looking on to, I think, probably our last question now, or pretty much our last question. We've only got a few minutes left. So a scientist investigates glucose production in swede plants. So when we're talking about glucose production in swede plants, hopefully immediately you think of photosynthesis, all right? Photosynthesis, because it's glucose production. He looks at 5 processes, A to E, that are involved in sugar production. He finds out how energy is lost in each process. So here's the energy is lost. I go around highlighting, as you can see, I'm underlining a lot of the parts of the question. It's really useful to take a highlighter into your exam and use it, not to don't highlight your answers, but to highlight the questions to keep you on track and to bring out the important words to you. All right. So it is useful to do, and I do encourage it. So it goes, sunlight hits chloroplast in the cells in the swede leaves, but then some of the energy is lost because some of that light energy is absorbed by cells without chloroplast, because it will be absorbed by their cell walls and other organelles, and that's no good because it's not going to be used for photosynthesis unless it hits the chlorophyll in the chloroplast. So some of it will be lost that way. Light is absorbed by photosynthetic pigments. Well, some energy is lost there because depending on the wavelength of the energy of the light, so it could be a different colored light, it could be the wrong wavelength of light to be absorbed, all right? Because obviously we have a broad spectrum of wavelengths in light. Then water molecules are split. Don't worry, you don't need to know about that process, but they're telling you, obviously energy is lost in that reaction. Then glucose molecules are produced, but there are limiting factors in this reaction. So not all the energy will be able to be used for this. And then glucose is stored in cells of Swede. And so some of that is lost in respiration. All right. So some of that is lost in respiration. And so, because some of it's obviously going to be used in respiration, isn't it? Some of that glucose is going to be used in respiration. So which process, A, B, C, D, or E, produces oxygen gas? So we've got to find out which bit do you think is going to produce waste oxygen gas? A, B, C, D, or E. So this is a slightly trickier question. Anybody got any idea what could be? Yes, brilliant. Okay, well done. Yes, it's C. Okay, yeah, it's A, when sunlight hits chloroplasts in cells. Oxygen is the waste product of photosynthesis. So it's the gas that's released at the end, and water is made up of H2O. So when that is split, we end up with hydrogen and oxygen being released. So well done with getting the C correct, because water is split into hydrogen and oxygen. How does the structure of a plant leaf help to reduce the loss in process A? So process A is the absorption of that sunlight energy, all right? And it's absorbed by cells without chloroplasts. So how are we stopping cells from absorbing that sunlight energy? Well, where are the most of the photosynthetic cells found? Well, they're found at the top of the leaf, aren't they? So palisade cells, brilliant. So we've got the palisade cells, which have got the most chloroplasts. The palisade cells are at the top of the leaf. They've only got the epidermis above them as far as cells are concerned. All right. And so because they're at the top, very few cells have to be passed through for that light energy to get to them. Brilliant. Okay, so all you have is the upper epidermis above them. So it's only one layer of cells above the palisade cells. So that is reducing how many cells are above them. So that's why the palisade cells are at the top. Excellent, well done. And then we look at the next part, carotene and xanthophyll. Now, some of you might not have heard of these before. Carotene and xanthophyll are a type of photosynthetic pigments. You've heard of chlorophyll. Chlorophyll is the green one. Well, carotene and xanthophyll are the orange and yellow ones that you see in autumn. So they help to reduce the energy loss in process B. Explain how they do so. So if they're a different colour, what's going to happen with the coloured light that they absorb? So chlorophyll is green because it reflects green light. Carotene and xanthophyll are orange because they reflect orange light. So if you have carotene and xanthophyll as well as chlorophyll all together in a chloroplast, it means that more light can be absorbed. So they absorb different wavelengths of light, that's one mark, so that a wider range or wider spectrum of wavelength can be absorbed. Okay. So wavelengths, so wider range of wavelengths. So wavelengths, different colours of light have different wavelengths. So you've probably done that in physics, I'm guessing. And so those different wavelengths have to be absorbed by different types of pigments. So we have the carotene and xanthophylls helping the chlorophyll. And I'm afraid that's all we have time for. I hope you've really enjoyed this lesson. Don't forget about the Tutor Online. Go and try signing up for your 2 weeks free of the GCSE tutoring platform. And I really hope that I'm going to be able to see you in some more of my lessons. Okay, goodbye and have a good rest of week. And I hope to see some of you next week. Take care. When does the code expire? I think that I'm not sure when it expires because it was 2 weeks starting this week, I think. Thank you. My pleasure. Brilliant. Okay, take care, everybody. It expires on Tuesday, 9th of March. Okay, off I go.